There is fitness advice that has become the foundation of healthy aging guidance. Exercise regularly to stay strong and energetic. Women over 60 dutifully walk, swim, cycle, and attend fitness classes, trusting that movement will preserve the cellular energy factories that power every function in their bodies. They complete their workouts, feel the satisfying fatigue of effort, and believe they have done what is necessary to maintain their vitality. Yet despite consistent exercise, their energy continues to decline, their stamina fades. The mitochondria that should be multiplying and strengthening seem to be responding with only a fraction of the adaptation they expected. Here is the counterintuitive insight. For many senior women, exercising alone cannot effectively build new mitochondria, no matter how consistent the routine or how appropriate the intensity. Exercise provides the signal that tells your cells to create more mitochondria. But that signal must be followed by specific nutrients that support the actual construction process. Without these nutrients present in the hours after exercise, the signal fades before construction can complete. Your body receives the instruction to build, but lacks the materials and cofactors to execute the blueprint. Here is what the research shows. Mitochondrial biogenesis. The process of creating new mitochondria requires the activation of specific genetic pathways followed by the synthesis of thousands of proteins and lipids that form mitochondrial structure. Exercise activates these pathways, but the construction phase depends on nutrient availability during the hours that follow. Studies demonstrate that subjects who consume specific nutrients after exercise show significantly greater increases in mitochondrial markers than those who exercise with inadequate post. Exercise nutrition. The exercise was identical. The mitochondrial outcome was profoundly different. What is happening in your cells when you exercise without proper post? Exercise nutrition involves incomplete execution of a building program. Exercise activates a master regulator called PGC1 alpha, which initiates the cascade of gene expression needed for mitochondrial biogenesis. This activation peaks during and shortly after exercise, creating a window when cells are primed to build new mitochondria. If the necessary nutrients are not present during this window, the building program runs without adequate resources. Some construction occurs, but far less than the activated program could have produced. In this talk, we will explore a simple but powerful upgrade to the way you support your exercise routine. A specific food consumed after exercise that research shows can amplify mitochondrial biogenesis by providing the compounds your cells need during the critical post. Exercise construction window. You will learn why exercise alone falls short, how mitochondrial building actually works, and a practical protocol you can implement after your next workout to finally achieve the cellular energy improvements you have been exercising toward. To understand why exercise alone cannot effectively build new mitochondria in aging adults, we need to examine how mitochondrial biogenesis actually works and what factors determine whether the exercise signal translates into actual mitochondrial construction. The biology of cellular energy adaptation is more complex than simply moving your body, involving specific molecular pathways that require specific support. Here is what the research shows. Mitochondria are the organelles responsible for producing ATP, the energy currency that powers virtually every cellular process. Each cell contains hundreds to thousands of mitochondria depending on its energy demands. Muscle cells, heart cells, and brain cells contain particularly high mitochondrial densities because of their intense energy requirements. The number and quality of your mitochondria directly determine your energy capacity, endurance, and metabolic health. Mitochondrial biogenesis is the process by which cells create new mitochondria. Unlike most cellular structures, mitochondria have their own DNA and reproduce through a process similar to bacterial division. However, most mitochondrial proteins are encoded in the nuclear DNA and must be synthesized, transported, and assembled in a coordinated process. This complexity means that mitochondrial biogenesis requires both the genetic signal to begin and the raw materials to complete construction. What is happening at the molecular level when you exercise involves activation of cellular energy sensors, Exercise depletes ATP and increases the ratio of AMP-TP in working muscles. This shift activates an enzyme called AMP-activated protein kinase, or AMK functions as a cellular fuel gauge that responds to energy depletion by triggering adaptations that will increase future energy capacity. One of its primary targets is PGC, 1-alpha, the master regulator of mitochondrial biogenesis. Studies published in the Journal of Physiology have documented the time course of PGC1-alpha activation following exercise. Research shows that PGC1-alpha messenger RNA increases dramatically within hours of exercise, peaks at approximately 3 to 6 hours post-exercise, and returns to baseline within 24 hours. This temporal pattern defines a window when cells are maximally primed for mitochondrial construction. What you provide during this window dramatically influences how much construction actually occurs. Think of exercise as placing an order for new mitochondria with a cellular construction company. The order is received and the work crews are mobilized. But the construction company needs materials delivered to complete the job. If materials arrive during the mobilization window, construction proceeds efficiently. If materials arrive late or not at all, the crews eventually disperse and the construction order expires partially unfulfilled. Each workout places an order. Each post-workout period determines how much of that order gets completed. As women age, the importance of this post-exercise window increases substantially. First, the exercise signal itself may be somewhat blunted in older adults, meaning the construction order is already smaller. Second, baseline mitochondrial function declines with age, making each successful addition more valuable. Third, the anabolic response to all stimuli tends to weaken with age, requiring more precise support to achieve adaptation. Mature bodies need more intentional support to achieve the same mitochondrial benefits that younger bodies achieve automatically. Research in gerontology has documented age-related changes in mitochondrial biogenesis capacity. Studies show that older adults show reduced PGC1-alpha activation in response to exercise compared to younger adults. However, research also demonstrates that this blunted response can be partially overcome with appropriate nutritional support. The aged cellular machinery is still capable of building mitochondria. It simply requires stronger signals and better supported construction conditions. There is additional research demonstrating that the quality of newly built mitochondria depends on the conditions present during construction. Mitochondria assembled under optimal nutritional support show better membrane integrity, more efficient electron transport chains, and greater resistance to future oxidative damage. The construction conditions affect not just how many mitochondria get built, but how well those mitochondria function once complete. What is happening at the structural level when mitochondria are built with proper support involves better incorporation of quality components. Mitochondrial membranes contain specific phospholipids that affect their fluidity and function. The electron transport chain proteins must fold correctly and assemble in proper orientations. Antioxidant protection during assembly prevents oxidative modifications that would impair these delicate structures. Each aspect of construction quality contributes to the final function of the completed mitochondria. Studies in the journal Mitochondrion have examined the functional characteristics of mitochondria built under different nutritional conditions. Research shows that subjects with higher antioxidant and polyphenol status during training periods develop mitochondria with superior respiratory capacity and lower rates of reactive oxygen species production. The number of new mitochondria may be similar. The quality and efficiency of those mitochondria differs based on the construction environment. Have you noticed that despite consistent exercise, your energy levels continue to decline, and your stamina does not improve as you expect? This frustrating pattern often reflects incomplete mitochondrial biogenesis. The exercise signal is being sent. The construction is not completing. Your cells receive the instruction to build but cannot fully execute without the specific nutrients that support the post-exercise construction window. There is another dimension to mitochondrial biogenesis that involves protection of existing mitochondria while new ones are being built. Exercise generates reactive oxygen species that can damage mitochondrial components. If this oxidative stress overwhelms cellular defenses, it can actually harm mitochondria even as the body attempts to build new ones. Antioxidant support during the post-exercise period protects existing mitochondria while the construction of new ones proceeds. Studies in the journal Free Radical Biology and Medicine have examined oxidative stress markers following exercise in older adults. Research shows that while exercise-induced reactive oxygen species are part of the adaptation signal, excessive oxidative stress can impair mitochondrial function and biogenesis. The balance between beneficial signaling and harmful damage depends partly on antioxidant availability. Appropriate post-exercise nutrition helps maintain this balance in favor of adaptation. Research published in the American Journal of Physiology has examined how post-exercise nutrition affects mitochondrial outcomes. Studies comparing subjects who consumed specific nutrients after exercise versus those who did not show significant differences in mitochondrial enzyme activity, mitochondrial DNA copy number, and measures of oxidative capacity. The exercise stimulus was constant. The nutritional support variable determined whether that stimulus produced maximal adaptation. Now that you understand that mitochondrial biogenesis requires both the exercise signal and post-exercise nutritional support, the question becomes clear. Why does exercising alone, without consuming specific nutrients during the construction window, fail to produce the mitochondrial improvements that aging women need? Here is what the research shows. The construction of new mitochondria requires specific molecular building blocks and cofactors that may not be adequately available in the post-exercise period without intentional nutritional support. Mitochondrial membranes require particular fatty acids. Mitochondrial enzymes require specific vitamins as cofactors. The synthesis of new mitochondrial proteins requires amino acids. The protective systems that shield constructing mitochondria from oxidative damage require antioxidants. A gap in any of these resources limits how much construction can complete. The post-exercise period presents particular challenges for nutrient availability. Exercise increases blood flow to working muscles while potentially reducing blood flow to the digestive system. Existing nutrient stores may be depleted by the exercise itself. The body's priorities shift toward recovery processes that compete for available resources, without specific attention to post-exercise nutrition. The cellular environment may not support optimal mitochondrial construction, despite adequate signaling. Think of it like having a building permit approved, but the construction materials stuck in traffic. The permit is valid. The construction crews are ready, but without materials on site, the building cannot go up. As time passes, the permit eventually expires, the crews move on to other jobs, and the opportunity is lost. Post-exercise nutrition is about getting the materials to the site while the permit is still active and the crews are still assembled. A simple tool to understand is the concept of the anabolic window for mitochondria. Just as muscle protein synthesis has a window of enhanced sensitivity, after exercise, mitochondrial biogenesis has a similar window when the cellular machinery is primed and receptive. Providing appropriate nutrients during this window produces outsized effects compared to providing the same nutrients at other times. The timing matters because it aligns nutrient availability with cellular readiness. Studies comparing post-exercise nutrition timing have documented this window effect. Research shows that nutrients consumed within 2 hours of exercise produce greater mitochondrial adaptation than the same nutrients consumed 6 or more hours later. The cellular signaling that creates the opportunity gradually fades. Early provision of nutrients catches the window while it remains fully open. Delayed provision catches only the closing edge or misses it entirely. Have you ever noticed that some periods of consistent exercise produce noticeable energy improvements, while other periods produce minimal adaptation despite similar effort? The variable is often what happened after the exercise. Periods when post-exercise nutrition was appropriately timed and composed produced adaptation. Periods when post-exercise nutrition was neglected produced limited results. The exercise provided the opportunity. The nutrition determined whether that opportunity was captured. Another factor limiting exercise only approaches is the specific nature of mitochondrial construction requirements. Mitochondria have unique structural features that require particular nutrients. Their double membranes contain specific lipid compositions. Their electron transport chain components require multiple vitamins as cofactors. Building new mitochondria is not just about general energy and protein availability, but about the specific molecules that mitochondria incorporate into their structure. Research in nutritional biochemistry has identified the specific nutrients most critical for mitochondrial biogenesis. Studies show that omega-3 fatty acids become incorporated into mitochondrial membranes. B vitamins serve as cofactors for numerous mitochondrial enzymes. Certain polyphenolic compounds activate the same signaling pathways that exercise activates, potentially amplifying the biogenesis signal. These specific nutrients have outsized effects on mitochondrial outcomes compared to general caloric or protein intake. The solution to maximizing mitochondrial biogenesis from your exercise routine is to consume polyphenol, rich berries, particularly blueberries or tart cherries, within 2 hours after completing your workout. These foods contain compounds that amplify the same cellular signaling pathways that exercise activates, extending and strengthening the mitochondrial biogenesis signal while providing antioxidant protection that allows construction to proceed without oxidative interference. Here is what the research shows. Polyphenolic compounds found in berries, including anthocyanins, quercetin, and resveratrol, like molecules, activate AMK and PGC-1-alpha through mechanisms similar to exercise itself. When you consume these compounds after exercise, you are essentially adding to the signal that exercise initiated. The combined activation produces a stronger and more sustained biogenesis response than exercise alone. Studies published in the Journal of Nutritional Biochemistry demonstrate that polyphenol supplementation enhances exercise, induced mitochondrial adaptations in both animal models and human subjects. What is happening when you consume berries after exercise involves synergistic activation of the mitochondrial biogenesis pathway. Exercise activates AMK through energy depletion. Berry polyphenols activate AMK through direct molecular interaction. The combined activation exceeds what either stimulus achieves alone. Additionally, the polyphenols activate sirtuins, another family of enzymes that promote mitochondrial biogenesis and longevity-associated pathways. The berry compounds essentially tell your cells that the exercise message should be heard even more loudly. Think of it like having two people push a heavy object rather than one. Exercise provides one push. Berry polyphenols provide another push in the same direction. The object moves further with combined effort than with either push alone. For mitochondrial biogenesis, the combined signaling from exercise plus polyphenols produces greater construction activity than exercise alone, even vigorous exercise. The antioxidant properties of berries provide additional benefit during the post-exercise construction period. Building new mitochondria requires protection from oxidative damage that could harm the delicate assembly process. Berry anthocyanins neutralize reactive oxygen species, creating a safer environment for mitochondrial construction. The same compounds that amplify the building signal also protect the building process. Here is what the research shows about specific berry effects. Blueberries have been extensively studied for their effects on mitochondrial function and biogenesis. Research published in the journal Nutrition and Metabolism found that blueberry supplementation increased mitochondrial biogenesis markers in muscle tissue and improved exercise performance. Tart cherries have shown similar effects, with studies demonstrating enhanced recovery and mitochondrial enzyme activity in subjects consuming tart cherry juice after exercise. Both fruits provide the polyphenolic compounds that support mitochondrial adaptation. Research examining the combination of exercise plus berry consumption has directly measured mitochondrial outcomes. Studies show that subjects combining regular exercise with daily berry intake show greater increases in citrate synthase activity, a marker of mitochondrial density, than subjects doing the same exercise without berry supplementation. The studies controlled for exercise volume and intensity. The berry consumption was the differentiating factor. Greater mitochondrial markers indicated more successful biogenesis. There is additional research demonstrating that berry polyphenols support mitochondrial function through multiple mechanisms beyond biogenesis signaling. Anthocyanins accumulate in mitochondrial membranes, where they protect against lipid peroxidation. Quercetin and related compounds support the electron transport chain function, improving ATP production efficiency. These functional benefits compound with the increased mitochondrial numbers to produce substantial improvements in overall energy capacity. What is happening in existing mitochondria when berry polyphenols are present involves protection and optimization of current function. You do not have to wait for new mitochondria to be built before experiencing some benefit. The polyphenols begin supporting your existing mitochondrial population immediately, while the longer-term biogenesis effects develop over weeks. This dual action, protecting current mitochondria while building new ones, accelerates the timeline for noticeable energy improvements. Studies in the Journal of Agricultural and Food Chemistry have traced the distribution of berry polyphenols after consumption. Research using labeled compounds shows that anthocyanins and other berry polyphenols accumulate preferentially in metabolically active tissues, including muscle and brain. Within cells, they concentrate in and around mitochondria, where their protective effects are most valuable. This targeted distribution makes berries particularly effective for mitochondrial support, compared to supplements that may not achieve the same tissue concentration. The polyphenols in berries also support mitochondrial quality through a process called mitophagy, the selective removal of damaged mitochondria. Healthy mitochondrial populations require both the creation of new mitochondria and the removal of dysfunctional ones. Berry compounds promote this quality control process, ensuring that your mitochondrial population improves not just in quantity, but in overall function. The result is a more efficient energy production system. Ask yourself this question: how many exercise sessions have you completed expecting to build mitochondria and improve your energy, only to see minimal adaptation despite consistent effort? The exercise was providing the signal. The post-exercise environment was not supporting the construction. By adding berries after exercise, you finally provide both the amplified signal and the protected construction conditions that maximize mitochondrial biogenesis. Now that you understand why consuming polyphenol-rich berries after exercise dramatically enhances mitochondrial biogenesis, it is time to learn exactly how to implement this protocol for maximum benefit. The details of berry type, amount, timing, and preparation all influence your results. Here is the first principle. Consume the berries within 2 hours after completing your exercise session. This timing captures the peak of the post-exercise construction window, when PGC1 alpha activation is highest and cells are most primed for mitochondrial biogenesis. Earlier within this window is generally better than later. Many women find that consuming berries within 30 to 60 minutes post-exercise works well with their schedules and appetite. The amount of berries that provides meaningful benefit is approximately 1 cup of fresh or frozen berries, or 8 ounces of tart cherry juice. This serving provides sufficient polyphenolic compounds to produce the signaling and protective effects research has documented. Larger amounts are not harmful but do not appear to provide proportionally greater benefit. Consistency of daily post-exercise consumption matters more than occasional large doses. Here is what the research shows about berry type selection. Blueberries provide particularly high concentrations of anthocyanins and have extensive research supporting their mitochondrial effects. Tart cherries, available as whole fruit, dried fruit, or juice, provide similar polyphenolic compounds with additional benefits for recovery and sleep. Blackberries, raspberries, and strawberries also contain relevant polyphenols, though in somewhat lower concentrations. Any of these options provides benefit. so choose based on preference, availability, and budget. A simple tool to apply is the smoothie method. Blend 1 cup of frozen berries with a liquid base such as water, milk, or plant milk immediately after your workout. Frozen berries blend smoothly, and the cold temperature can be refreshing post-exercise. The blending releases polyphenolic compounds from the berry cell walls, potentially improving absorption. Adding a handful of spinach or a scoop of protein powder creates a more complete post-exercise recovery beverage without diminishing the berry benefits. The form of berry consumption affects polyphenol delivery somewhat. Fresh and frozen, whole berries provide fiber alongside polyphenols, supporting gut health and moderating sugar absorption. Juices like tart cherry juice provide concentrated polyphenols without fiber, which may be preferable for those with digestive sensitivities. Dried berries provide convenience but contain concentrated sugar and may have some polyphenol loss. During processing, all forms provide benefit, so choose based on practical considerations. Here is what the research shows about consistency. The mitochondrial benefits of berry consumption accumulate over time through consistent practice. Studies following subjects for 8 to 12 weeks show progressive improvements in mitochondrial markers with continued berry consumption after exercise. Single doses provide some benefit, but the cumulative effect of daily post-exercise berry consumption produces substantially greater adaptation. Make this a permanent part of your exercise routine, not an occasional addition. Have you noticed that nutrition habits you enjoy are the ones you maintain long-term? Berries are naturally sweet and enjoyable for most people. The post-exercise smoothie or berry snack can become something you look forward to, a reward for completing your workout that also happens to support your mitochondrial health. When the habit is pleasant, consistency becomes easy. A practical framework for implementation involves preparing your post-exercise berries before you exercise. Set up your smoothie ingredients so you can blend immediately upon returning. Have tart cherry juice in the refrigerator ready to pour. Place a container of fresh berries where you will see them after your workout. Reducing friction between exercise completion and berry consumption helps capture the optimal timing window. Here is what the research shows about interactions with other post-exercise nutrition. Berries can be combined with other recovery foods without diminishing their mitochondrial benefits. Consuming berries alongside protein supports both mitochondrial and muscle adaptation. Consuming berries with carbohydrates replenishes glycogen while providing polyphenols. There is no need to consume berries in isolation. Integrate them into whatever post-exercise eating pattern works for your overall nutrition goals to make the post-exercise berry protocol work even more powerfully for mitochondrial biogenesis. Consider the additional factors that influence how effectively your body can build new mitochondria. Exercise type, sleep, overall nutrition, and stress all affect mitochondrial adaptation. Here is what the research shows. Different exercise types provide different strengths of mitochondrial biogenesis signal. Endurance exercise like walking, cycling, and swimming provides particularly strong activation of the PGC-1-alpha pathway. Higher intensity intervals can produce even stronger activation in shorter time periods. Resistance exercise provides some mitochondrial stimulus alongside its muscle-building effects. Any exercise provides benefit, but endurance and interval training may be particularly synergistic with post-exercise berry consumption for mitochondrial outcomes. The combination of aerobic challenge plus polyphenol support creates optimal conditions for cellular energy adaptation. A simple tool to apply is including some endurance or interval component in your exercise routine. Even 10 to 15 minutes of sustained moderate intensity movement or several short high-intensity intervals provides meaningful mitochondrial stimulus. Combined with post-exercise berries, this exercise approach maximizes your mitochondrial biogenesis potential. If your current routine is entirely resistance-based, consider adding a cardio component specifically for mitochondrial benefits. Sleep profoundly affects mitochondrial biogenesis and repair. During sleep, growth hormone release supports anabolic processes including mitochondrial construction. Sleep also allows completion of repair processes that protect existing mitochondria from accumulated damage. Research shows that sleep deprivation impairs exercise adaptations, including mitochondrial biogenesis. Prioritizing 7 to 8 hours of quality sleep supports your body's ability to execute the mitochondrial construction program that exercise and berries initiate. Here is what the research shows about overall antioxidant status. While berries provide concentrated polyphenols post-exercise, your overall dietary antioxidant intake affects baseline cellular protection significantly. A diet rich in colorful vegetables, fruits, nuts, and other plant foods provides diverse antioxidants that support mitochondrial health throughout the day. The post-exercise berries provide targeted support during the critical construction window. The broader antioxidant intake provides ongoing protection that complements this targeted approach. B vitamins serve as essential cofactors for mitochondrial enzymes. Deficiency in any B vitamin can impair mitochondrial function, regardless of how well the biogenesis signal is supported. Ensuring adequate B vitamin intake through diet or supplementation provides the cofactors that newly constructed mitochondria will need to function properly. The berries support the building process, while the B vitamins support the operation of what gets built, creating a complete support system for mitochondrial health. A practical framework for comprehensive mitochondrial support involves the post-exercise berries as the anchor, surrounded by a foundation of adequate sleep, diverse dietary antioxidants, and sufficient B vitamins. Each element supports the others. The berries provide the targeted post-exercise boost. The foundation provides the overall environment where that boost can produce maximum results. Chronic stress impairs mitochondrial biogenesis through elevated cortisol and altered energy allocation. Research in cellular biology demonstrates that stress hormones shift cellular resources away from long-term adaptation toward immediate survival responses. Managing stress through relaxation practices, time in nature, social connection, and adequate rest creates an internal environment more favorable to mitochondrial construction. The connection between psychological state and cellular energy production reflects how deeply our mental and physical systems are interconnected. Have you noticed that your energy and adaptation seem better during relaxed periods of life compared to stressful periods? This observation reflects stress hormones interfering with mitochondrial biogenesis and repair. By managing stress alongside your exercise and nutrition protocol, you remove a break that would otherwise limit your mitochondrial adaptation. Understanding the science creates the foundation, but lasting mitochondrial improvement requires consistent action maintained over the months needed for adaptation to accumulate. This is where behavioral psychology becomes essential for success. Here is what the research shows. Human behavior is governed by 3 primary forces. Motivation fluctuates and cannot sustain action alone. Ability depends on how simple and convenient the behavior is. Prompts are the cues that trigger the behavior at the right moment. For a post-exercise nutrition protocol to become permanent, it must be easy to execute, connected to existing routines, and rewarding enough to maintain. A powerful tool to apply is pairing the berry consumption with exercise completion. The end of your workout becomes the cue for berry consumption. You do not need to remember separately. You do not need to decide each time. Exercise ends, berries follow. The connection becomes automatic with repetition, requiring no ongoing willpower or decision. Habit stacking connects the berry consumption to your existing post-exercise routine. Perhaps you already drink water after exercise. Add berries to that water in smoothie form. Perhaps you already have a snack after exercise. Make that snack berry based by integrating berries into existing post-exercise behaviors. You minimize the new habit formation required. The enjoyable taste of berries supports adherence. Unlike many health interventions that require tolerating unpleasant experiences, berry consumption is intrinsically pleasant for most people. The natural sweetness, the refreshing quality after exercise, the variety of preparation options all contribute to a positive experience that reinforces the behavior. You are more likely to continue a practice you enjoy. Have you noticed that the health habits you maintain long-term are the ones that feel rewarding rather than punishing the post? Exercise berry protocol works partly because it feels like a treat rather than a chore. The berries are delicious. The smoothie is refreshing. The habit fits naturally into the satisfaction of completing a workout. When the behavior feels good, consistency follows naturally. Tracking provides feedback during the weeks before energy improvements become dramatic. Notice how your recovery feels after workouts. Notice whether your energy levels during the day seem more stable. Notice whether your endurance during exercise sessions improves over time. These functional markers often precede dramatic subjective changes and provide encouragement during the adaptation period. When you bring together everything we have explored, a clear and empowering picture emerges. Exercise provides the essential signal that initiates mitochondrial biogenesis, but that signal must be supported by post-exercise nutrition to translate into actual mitochondrial construction. Without appropriate nutrients during the construction window, much of the exercise signal fades before new mitochondria can be built. Polyphenol, rich berries consumed after exercise provide the ideal support for the post-exercise construction window. Their polyphenolic compounds amplify the same signaling pathways that exercise activates, strengthening and extending the biogenesis signal. Their antioxidant properties protect the construction process from oxidative interference. Their natural palatability supports the consistent consumption needed for cumulative benefits. You now understand why years of consistent exercise may have produced disappointing improvements in energy and stamina. The exercise was providing the signal. The post-exercise environment was not supporting the construction. When you add berries after exercise, you finally provide both amplified signaling and protected construction conditions that maximize mitochondrial biogenesis. You have learned that the protocol is simple. Consume approximately 1 cup of berries or 8 ounces of tart cherry juice within 2 hours after completing exercise. Maintain this practice consistently with every exercise session. Allow 8 to 12 weeks for cumulative mitochondrial adaptation to produce noticeable energy improvements. You have seen that supporting factors amplify these benefits. Including endurance or interval components in exercise strengthens the biogenesis signal. Quality sleep supports construction and repair. Overall dietary antioxidants provide background protection. Stress management removes hormonal brakes on adaptation. Each factor contributes to the environment where your post-exercise berries can work most effectively. Perhaps the most important insight is that exercise adaptation is not automatic. It requires both the signal and the support. Berries provide the support that transforms your exercise signal into actual mitochondrial construction. When you consistently pair exercise with berries, you finally capture the full adaptation potential of every workout. Here is your challenge for your next exercise session. Complete your workout, then consume 1 cup of berries or a berry smoothie within 2 hours. Notice how your recovery feels. Make this pairing your standard practice for every exercise session going forward. Over the coming months, observe the cumulative improvements in your energy, endurance, and overall vitality. Your mitochondria have been waiting for the support to fully respond to your exercise efforts. The signal was arriving. The construction conditions were incomplete. Berries provide the amplification and protection that finally allow your exercise to produce the mitochondrial adaptation it should have been producing all along. Aging does not mean accepting declining energy and stamina. It means providing your cells with the specific support they need to adapt and improve. When you pair exercise with post-exercise berries, you create the conditions for mitochondrial biogenesis that restore the cellular energy capacity underlying vitality at any age. If this guidance brought clarity or a sense of empowerment, I invite you to like this video and share your experiences in the comments. Let us know how your energy responds to the Exercise Plus Berries protocol. Subscribe to join a community dedicated to science-driven, practical strategies for health and vitality at every age. Your mitochondrial biogenesis is waiting to be fully supported. Your next workout is the moment to begin.